Good morning, everyone, and welcome to the first PGC virtual conference. OnePGC, PGC in Luzon, Visayas, and Mindanao, para sa bayan. To officially open the program, we would like to introduce our conference host, Mr. Carlo M. Lapid, a senior bioinformatics specialist from PGC's core facility for bioinformatics.
Good morning, everyone, and welcome to the first PGC virtual conference, OnePGC, PGC in Luzon, Visayas, and Mindanao, hashtag para sa bayan. PGC stands for the Philippine Genome Center, a research and service unit of the University of the Philippines located at UP Diliman. Founded in 2009, the institution now has core facilities in DNA sequencing and bioinformatics, clinical genomics, protein, proteomics, and metabolomics, as well as research and development programs in health, agriculture, livestock, fisheries and forestry, biodiversity, forensics and ethnicity, and computational genomics and systems biology. Before we proceed, we'd like to thank each and every one of you for taking the time to attend this online gathering, which will give recognition to our partners and stakeholders from Luzon, Visayas, and Mindanao. These people and institutions help in the national call for increased capacity for COVID-19 testing in the country, and for that we thank them. 2020 has brought upon us a lot of challenges, and we do hope that everyone is in good health and taking the necessary precautions to stay safe during this pandemic. For this event, we have invited various government agency representatives, as well as some of PGC's very own experts and program directors, to give messages and updates, all related to PGC's efforts in response to COVID-19. To begin, let us welcome the Executive Director of the Philippine Genome Center, Dr. Cynthia P. Saloma, to give the welcoming remarks for today's event.
Department of Health Secretary Francisco Duque III, DOST Secretary Fortunato Boy de la Peña, Commission on Higher Education Chairperson Prospero Popoy de Vera III, UP President Danilo Concepcion, DOH Assistant Secretary Nestor Santiago, DOST Undersecretary Rowena Guevara, UP Vice President for Academic Affairs, Cynthia Rose Banzon Bautista, officials of various local government units, PGC Execo members past and present, the entire PGC family in Luzon, Visayas, and Mindanao, volunteers, frontliners, partners in the public and private sectors, research collaborators, guests, friends in the media, good morning po, and allow me to welcome you all today. For some of us, the next few days will not be a countdown for Christmas Eve, but more of counting the days to finally bid farewell to 2020, a pivotal year in our nation's history and in the history of humankind. While we are grateful for being given the chance to focus on our health and family, we can't help but feel anxious for what COVID-19 brought upon us collectively. This pandemic is a wake-up call not only to us Filipinos, but to everyone the world over. On January 30, 2020, PGC released a statement on our website that the Philippine Genome Center gears up to verify presence of 2019 nCoV through whole genome sequencing. And at that time, not one of us had an inkling of that we will be working side by side with the Philippine Department of Health against the SARS-CoV-2 outbreak. From then to today, the Philippine Genome Center, through its clinical genomics laboratory, has tested more than 40,000 unique individuals for COVID-19 using the RT-PCR method, while PGC Visayas as well as PGC Mindanao have trained more than 100 health workers and frontliners on molecular techniques to capacitate the hospitals and molecular diagnostic laboratories in the regions in getting accreditation for COVID-19 diagnostic testing. Perhaps you may be wondering why we are holding this conference when we are already so busy. We are organizing this virtual conference, PGC Luzon, Visayas, and Mindanao #Para sa Bayan, to celebrate teamwork and bayanihan, because we believe that we can get through this crisis together, banking on our various institutions' individual strengths to rise above the pandemic. The talks and the accompanying photographs and videos today will document the extraordinary journey that we at the Philippine Genome Center and all of us as a community took together to face the coronavirus pandemic, knowing full well that we have to be united in this endeavor. This conference is our distinct opportunity to convey our sincerest appreciation to our donors, both in the private and public sectors, our teams in Luzon, Visayas, and Mindanao, our research collaborators, and our key guests today, UP President Danilo Concepcion, Chancellor Fidel Menenzo, Vice President Maria Cinteros Bautista, DOS Secretary Boy de la Peña, DOS Undersecretary and the ever supportive USEC Rowena Guevara, JED Commissioner Popoy de Vera, NIMBB Dilman Foundation President Francis Fuentes Acosta, and most of all, DOH Secretary Duque, who all believed and put their trust in our young team at the PGC. With all their support and your assistance, we were given the chance, this singular opportunity, to assist and contribute on this battle against COVID-19, which validates the very reason why the Philippine Genome Center was established in the University of the Philippines system 10 years ago, to prepare the country for pandemics, to provide scientific-based solutions to societal concerns, and to address the pressing needs of the country in health and agriculture. We hope that this conference brings to everyone a virtual pat in the back for trying our best to cope and adapt to the new normal. And we also hope that the data and the relevant information from the three presenters we highlight today, plus a peek at our 6-year strategic plan, will inspire you to look beyond the struggles of 2020 and move forward on a positive note in 2021. Maraming salamat pong muli at magandang umaga sa ating lahat.
Thank you very much, Dr. Saloma. And now to give us the first message, it is my honor to introduce to you the University of the Philippines President, Attorney Danilo L. Concepcion.
Magandang umaga po sa inyong lahat. On behalf of the University of the Philippines, I congratulate the Philippine Genome Center for today's event. After the successful online inauguration of the PGC Visayas last November 20, here we are again with OnePGC, PGC in Luzon, Visayas and Mindanao, #Para sa Bayan. I commend the PGC team for organizing this virtual conference, which emphasizes teamwork and bayanihan. These two qualities are crucial as the nation rises above the pandemic caused by COVID-19. As your president, I am very proud and appreciative of the collective efforts of our UP scientists and researchers and the various UP units who continue to work closely with the Department of Science and Technology, the Department of Health, and other government agencies in the fight against this pandemic. The PGC, especially throughout this year, has been an exemplar of the university's commitment to honor and excellence. And with the PGC UP Mindanao Satellite Facility, the first of its kind in Mindanao, and the PGC Visayas in Miagao, Iloilo, the Philippine Genome Center is better able to fulfill its mission to utilize advanced knowledge and emerging technologies in genomics and bioinformatics in health and medicine, agriculture, biodiversity, forensics and ethnicity, industry, and the environment for Filipinos. Like UP as the national university, PGC will continue to espouse bayanihan and unity in all its endeavors. Nagkakaisa po tayo para sa bayan. Muli, maalab na pagbati sa Philippine Genome Center at mabuhay tayong lahat.
Thank you very much, Attorney Concepcion, for those remarks. Our next speaker is a person whose patronage and support were pivotal in the establishment of the Philippine Genome Center. Let us all welcome the University of the Philippines Vice President for Academic Affairs, Dr. Maria Cynthia Rose Banzon Bautista.
DOST Secretary De La Peña, DOH Secretary Duque, CHED Chairman De Vera, President Concepcion, PGC Executive Director Cynthia Saloma, highly respected colleagues and friends in PGC. It is my honor and pleasure to greet you all on this auspicious occasion of a virtual conference. I have been extremely privileged to be working with you and observing from a distance your unstinting commitment and concrete contributions at the frontiers of genomics research, as well as your exemplary public service in this time of COVID. As PGC moves from strength to strength, let me use this occasion to convey our gratitude to the teams, donors, and PGC partners for enabling the Philippine Genome Center to conduct COVID-19 PCR testing. Let me also thank Dr. Maralit, Dr. Yap, and Dr. Ayes for sharing scientific insights that will help us deal more effectively with national concerns such as this pandemic that challenges not only the way we live, but as an educational institution, the way we teach, learn, and reach out to students of diverse backgrounds. Finally, we deeply appreciate the leadership of Dr. Cynthia Saloma at this critical juncture when it is imperative to carry out PGC's 6-year strategic plan and allow its implementation and the center's adjustments to the learnings from the COVID situation to further nuance and enhance the plan. Mabuhay ang PGC.
Congratulations to the University of the Philippines for the successful establishment of the Philippine Genome Center in Luzon, Visayas, and Mindanao. This is a very big milestone, not only for the University of the Philippines, but for the overall landscape of genomic research in our country. Undeniably, we have come a long way since we started our vision to tap genomics research for health, agriculture, livestock, fisheries, and biodiversity way back in 2009. In line with the challenges brought by COVID-19, we also highlight how we can leverage genomics research to address national concerns or emergencies. From genomic sequencing of COVID-19 cases to boosting our testing capacity, the Philippine Genome Center is one of the institutions at the forefront of our pandemic response. As exemplified in our ongoing battle against COVID-19, our very own researchers and experts are capable of delivering and producing innovative and world-class solutions. It is our commitment at the Department of Science and Technology to nurture our own pool of experts and to support their scientific and technological endeavors. In every field, not just in genomics, our work and our vision will always be directed to serve the needs of our people. Allow me to thank the University of the Philippines, the Philippine Genome Center, and our Consortium for Health Research and Development, our researchers, and everyone who has been our partner in our continuous work to advance science for the people. This is for a progressive research community for genomics, for science, for the Filipino people, and for our country. Maraming salamat po at mabuhay.
Thank you very much, Secretary de la Peña. And now our next guest is one of our champions who supported the expansion of our activities in Visayas and Mindanao. Without further ado, let us welcome DOST Undersecretary for Research and Development, Dr. Rowena Cristina Guevara.
Department of Health Secretary Francisco T. Duque III, Commission on Higher Education Chair Professor Prospero de Vera III, UP President Danilo Concepcion, UP Vice President for Academic Affairs Maria Cynthia Rose Banson Bautista, Philippine Genome Center Executive Director Dr. Cynthia Saloma, PGC Deputy Executive Director Dr. Raul Destura, members of the UP Diliman National Institute of Molecular Biology and Biotechnology Alumni Foundation, my colleagues from the Department of Science and Technology, PGC frontliners and support teams, speakers, guests, our friends from media, ladies and gentlemen, good morning. Magandang agham sa inyong lahat. It is with great pleasure and privilege that I am attending this event that aims to recognize the contributions of the Philippine Genome Center in our fight against COVID-19. The theme, PGC in Luzon, Visayas, and Mindanao, hashtag Para sa Bayan, is truly reflective of the PGC's mission to provide a deeper understanding and judicious application of advanced knowledge and emerging technologies in genomics and bioinformatics in health and medicine for the benefit of Filipinos throughout our country. Even before the pandemic, the DOSC has always recognized the importance of utilizing genomic technology platforms for the development of local technologies. Through the Philippine Council for Health, Research and Development, or PCHRD, DOST has spent more than 900 million pesos on various genomics programs, which includes the establishment of the PGCs in the country. These investments led not only to advances in genomic research, but also provided solutions as well to the country's clinically unmet needs, especially during this pandemic. Realizing that research and development has an essential role in the response to the COVID-19 outbreak, the PGC, through the support of DOST, has paved the way to the development of the Gen Amplify COVID-19 Detection Kit, which is the first locally developed COVID-19 detection kit that expedited the process and turnaround period for COVID-19 testing in the Philippines. In April 2020, the Research Institute for Tropical Medicine, or RITM, issued a certification allowing the PGC Clinical Genomics Laboratory to perform independent testing for COVID-19. PGC opened a drive-through and on-foot COVID-19 testing services for individuals without symptoms and required testing for clearance and other purposes. Recently, the center expanded these services to offer the test to a more affordable price compared to other hospitals and institutions, making it more accessible for people who want to get tested for COVID-19. PGC Visayas and Mindanao were able to conduct a series of training on biosafety and biosecurity, as well as RNA extraction and the use of Gen Amplify COVID-19 detection kit to capacitate them for COVID-19 testing. These developments led to the quick identification, treatment, and immediate isolation of cases that help prevent the spread of the virus all over the country. Currently, the PGC is implementing several DOST-PCHRD supported research, such as the biosurveillance of COVID-19 in the Philippines through the whole genome sequencing of SARS-CoV-2 from patients. Number two, field implementation study of a locally developed diagnostic kit for the detection of COVID-19. Number three, severe acute respiratory infection or SARI metagenomics program and portable sequencing platform for COVID-19 detection. And fourth, lab on a chip platform for SARS-CoV-2 detection and diagnosis. Ultimately, this research will play an important role in the bigger public health picture, especially in our mitigation efforts and in helping investigators characterize the prevalence, spread, and contagiousness of the disease. Indeed, DOST's investment in genomic research has proven to be a game changer in advancing genetic research and health systems response in the country amidst this global crisis. But let me emphasize that this is not a sole undertaking by the DOST. We are grateful for the collaborations and commitment of our partners, such as the Department of Health, the Commission on Higher Education, and the University of the Philippines. Without them, we will not be where we are today. Indeed, COVID-19 has imposed a lot of changes and threats against public health. But we hope that as we work together as one, we can also overcome all of these challenges together. Muli, maraming salamat po sa inyo at mabuhay po tayong lahat.
Thank you very much, Undersecretary Guevara. Next, we will be having our main talks for this morning's conference from three of the facility heads here at PGC, each of which will be followed by a brief Q&A. Before we proceed, allow me to go over some brief guidelines on how questions can be submitted and selected. We do strongly encourage all of you to take this opportunity to send your questions to our distinguished speakers as they share their expertise and provide updates regarding PGC's work on COVID-19. Yes, I'm sorry about that. So regarding our guidelines for submitting and selecting questions, selected questions will be posted on the screen after each presentation to be answered by the panelist or the corresponding speaker. If you'd like to submit a question, and we do encourage you to do so, simply click on the Q&A box below on Zoom and enter your question there. Regarding this live stream, this conference is being live streamed via the Philippine Genome Center's Facebook page at www.facebook.com/phgenome. For questions or immediate concerns regarding this conference, please reach out to the OnePGC team via capacitydevelopment@pgc.up.edu.ph. And now on to our first main speaker for this conference, the Program Director for PGC's DNA Sequencing Core Facility, Dr. Benedict A. Maralit. He will be giving a talk entitled Sequencing in the Pandemic, DSTF Sequencing Activities for COVID-19 Research.
So good morning, everyone. First, I'd like to send a shout out to my fellow scientists, researchers, and members of the academy for your continuous efforts to, respond to the pandemic while also trying to make amends with the restrictions in terms of doing research and coming up with research outputs during the pandemic. And I also like to thank them for bearing with the SCF as we diverted some of our energy into our own PGC initiatives to respond to the pandemic instead of, giving our usual services for research. So for this presentation, I'd like to highlight our thanks to our stakeholders as well as our allies in the academe. And so the title of my presentation is really about sequencing in the pandemic, the SCF's sequencing activities for COVID-19 research. So I'm pretty sure that you may have heard this, but we cannot emphasize this enough that PGC's reason for existence is actually responding to a pandemic such as COVID-19. So some time ago, a center has been envisioned to lead in the country's genomic surveillance efforts to manage highly relevant diseases that lead to pandemic. And so here we are, passionately stepping up, heeding the call of the Filipino people for improved health response and management of the ongoing pandemic. So let me just share with you some of the humble beginnings of the SCF as one of the first programs developed by PGC. So originally, I think it was around the time of 2011 when a capacity building program proposal was sent, reviewed, and approved by the OSTP SURE. The actual title of this proposal was Capability Building in R&D on Genomics, a proposal to establish, of course, the so-called now as Philippine Genome Center through the first facilities such as the DNA sequencing core facility and the bioinformatics core facility. So which is now called as core facility for bioinformatics. Also, I'd like to highlight that, sometime 10 years ago, our executive director of the Philippine Genome Center and executive committee members, including our current director, Dr. Cynthia P. Saloma, presented to the then President Gloria Macapagal Arroyo the rationale behind the creation of the country's own genome center. There, we posed the challenge, if there is going to be a pandemic, are we prepared? Right now, gladly, we would answer yes. And in 2011, as I mentioned, our vision was really to have a fully operational service facility from a virtual one, which was in NIMBB. And then, of course, we were envisioned to create a research, education, and training in genomics and bioinformatics. And just to show you a brief, or at least an overview of what was the vision during those times. So this is the diagram of the SCF and PGC's envisioned efforts and tasks would be. So, and here we are now, 10 years after, you would see that we now have a COVID testing facility, a service lab for offering genomic sequencing services, as well as trainings, and many other things that is relevant to the lives of, the common Filipino people. So I'd like to show you some of, or at least the current DSCF staff that help cater to the needs of our fellow scientists and operate daily in our facility. So these people here are also important to establishing the PGC's diagnostic facility for COVID-19. So DSCF has actually been the birthplace of PGC's COVID testing lab. And to note, at the start really of 2020, we were already conducting preliminary activities, meetings to establish the clinical genomics lab. So originally, the CGL was envisioned to be the translational arm of the PGC. And then due to the emergence of the current pandemic, we decided to divert some of our activities to establish the testing lab. So then we were able to prepare for ISO certification and then, of course, development of new products and services, which led to the testing facility. We were able to set up a BSL-2 equipped COVID diagnostic facility, and then actually we received our accreditation by the end of 2020. By the end of of April 2020. And this would have not been possible without, of course, the help of our dear friends from NIMBB, particularly. I remember there were times that, oh, we need something like this, a component for VTM, and then do you have something like this from your lab? And then we would go there and then pick up them as we needed them. And that's really a very important aspect of how the success of the establishment of the CGL came to be. And of course, without the help of the MBB Foundation, which actually shouldered a big part of the capital that we needed to establish the testing lab. And so I would also like to send our thanks to people other than MBB Foundation, such as those that gave their donations, help, whether it's monetary or whether it's provisions for food and then for quarters. So all of the people in the UP community as well as around Metro Manila, they came to PGC to lend a hand in establishing and have a lab, a diagnostic lab where we can actually contribute to increase the testing capacity as people were clamoring for massive tests during the first few months of the pandemic. So I'm just showing you here some of the things that or at least a detailed view of what were our expenses and what we needed to buy for establishing the lab. And most of these details have been published and you can actually see them in our website. So a big thanks to all of you who were really part of the Bayanihan spirit. So some notable activities during the accreditation period, meaning to say the DACF staff members are a big part of the operations of the testing facility. So as you know, sometime in January, we had an emergency meeting, and then 2 months after, in March, we were already talking with the UP COVID-19 response team to set up contingency measures on how we will be operating during the pandemic. And then, of course, with the help of our friends from RITM, we were able to talk about determine the layout of the lab that needs to be complied. And then, of course, our friends also from the national team for biosafety, we'd like to thank them as well for allowing us to visit their lab so that we could review, assess the risks, make sure that our lab is compliant with the biosafety guidelines of RITM, as well as ensuring that our staff is really safe from laboratory infections related to the testing activities that we'd like to do. And then, of course, sometime in April, we were able to submit the PT panel for accreditation, which led to eventually our certification as an independent testing lab. And so, getting that certification, we were able to start our operations for testing, which were mainly made possible by the current DSCF staff and some volunteers, around 15, from NIMBB. And then July 1 marked the new normal operations of the SCF, where we returned our usual sequencing services, offered them to our dear friends who would like to continue their research. So here are just some of the information that and statistics that showed the operations of the DSCF and CGL's transitionary phase. So for a span of 2 months and with 15 NIMBB volunteers and 9 DSCF personnel, we were able to process around 200 samples per day as an average. And then in those 2 months, we were able to process around 1,500 samples. And that's a big chunk already of our contribution to the need for at least increased testing capacity. So we were also involved in other UP initiatives through the SCF and an NIMBB SARS-CoV-2 testing hands-on training program. So that was held in April to May for a period of around 5 weeks that actually empowered, built capacities in 27 institutions through training of 118 lab personnel. And then, of course, there was also a need, and we determined that we needed to increase our scope and breadth of the training program. But of course, the bottlenecks would be the hands-on locations. So we moved this training program into the online platform. And so UP and NIMBB thought of, and UP in general thought of the UP Trains program that's composed really of three separate programs or trainings headed by the first one headed being by the UPNIH and TCBB, the Beat COVID training course for Biosafety, and then of course NIMBB and PGC's UP collab with the satellite facilities in Visayas and Mindanao, and then of course the hands-on training. So the UP collab in particular is already 20 weeks out of 24, and we were able to train around 1,000 institutions with more than 1,000 lab personnel. And indeed, we were generating around 800 lab personnel who are at least skilled and have the right knowledge for doing the molecular testing for COVID-19. Some other things that we did during our collaboration with UPPGH is through the field implementation of, the locally developed diagnostic kit for the detection of COVID-19. DSCF's genomic sequencing was actually used as a reference standard for detecting the SARS-CoV-2 viral RNA. So the capillary sequencing service was used as a positive reference standard, and then for metagenomic sequencing, that's a negative reference standard. Because of this, we were able to generate two field studies of the Gen Amplify kit. The first one would be in collaboration with PGC, DSCF, and UPPGH, and then of course the review that was done by RITM. And because of this, eventually the UP developed diagnostic kit was accredited and recognized by the Philippine FDA. So aside from those, we were able to do and generate other results from this collaboration. That is the optimization of in-house protocols for whole genome sequencing. If you remember, that's PGC Bulletin 1. We were able to generate baseline shotgun sequencing of around 250 samples, and then that in turn generated whole genomes from Philippine cases, essentially giving the Philippine representation into the worldwide repository of SARS-CoV-2 genomes. And then, of course, we were also able to detect the D614G variant that has been well talked about in the previous months through, of course, the use of next generation sequencing and capillary sequencing. So we did have results that are putative and then confirmed in the lab. We not only did projects and initiatives that were mainly focused on COVID-19, of course. We were also able to do some other things that were part of our new normal operations, which, as you know, started in July 1. So actually, the NGS projects for COVID-19 was just a third of what we were doing for the whole year. This is mainly to cater to the demands and the needs of our fellow colleagues in the research institutes that's needed to have research outputs by the end of the year. So we also have an upcoming project that is funded by the PCHRD to expand our biosurveillance efforts in PGC with regards to whole genome sequencing of the SARS-CoV-2. So our main research objectives would be to collect RNA samples and then sequence them and eventually complete SARS-CoV-2 genomes so that we can determine some epidemiological implications and then look at biosurveillance results so that we could come up with PGC bulletins that may be of relevance to development of new policies. So I think that covers most of our work for the SCF, and I think I could not thank you all enough for being with us and helping PGC with all our efforts to respond to the pandemic. Thank you very much for listening.
Thank you very much, Dr. Maranit, for that very informative and useful presentation. And now, to all our guests, we will be proceeding with a Q&A for Dr. Maranit's talk. Again, we'd like to encourage everyone to submit questions by clicking on the Q&A tab below on Zoom. So I believe we have at least a couple of questions that came in, if we could project that. Okay, so Dr. Moralit, one question for you. What other services are being developed or prepared by DSCF for 2021?
Okay, so we have prepared several things for 2021. So initially, or we have always been trying to improve the efficiency of our services. So in 2021, our end users can expect to have improved quality of life in terms of transacting with the SCF, email requests or requests for services, things like that. So we're trying to deploy our LIMS that has a front end and a back end in the lab. And then of course, also now that the clinical genomics lab has already been established, particularly that caters to the COVID testing, we're also now ready to at least explore other services such as tumor profiling, genetic services. So as early as the first quarter of 2021, we'll start to roll out and explore giving this or offering these services to the public. Thank you for that question.
Thank you very much, Dr. Marali. I believe we have at least one or two other questions, if we could have those projected. How does sequencing SARS-CoV-2 guide the country's public health efforts?
So as I mentioned, information on the SARS-CoV-2 genome has a lot of implications, more particularly in the biosurveillance, because what we are seeing is the evolution of the virus. So, and information from the evolution of the virus is related to finding out clusters, which ones are more closely related, which clusters are coming from what locality. So sequencing of the SARS-CoV-2 genome will help us determine if there is a super spreader, what might be the origin of the virus, and then we can pinpoint, okay, this is where we should focus our travel restrictions, or at least for lockdowns that are on high risk areas. I think that's one of the areas that might be used, that sequencing might be useful. And also that sequencing will also give us an idea whether our current testing detection methods are also up to date and still able to detect the virus, mainly by determining whether there is already a significant mutation that affects the detection kit.
Thank you again, Dr. Marali. That was very informative. I think we have time for one more question. So an attendee submitted this. What are the effects of the spike of D614G mutation in the viral pathogenesis and transmissibility regarding the prevalence of SARS-CoV-2? Actually, I think Dr. Ayas also might have insight regarding this question.
Yeah, I think we'll be having more of this, this more detailed discussion regarding this, what do you call this, popular mutation. But based on our readings, there is really no um, significant correlation between the existence of this mutation in relation to any clinical outcomes. So I guess the general idea, or I guess in the community, there is not much anxiety with regards to the presence of this mutation, because really there's nothing to worry about. There's no increased transmissibility. There's no disease severity outcome that may be related to this. But I think we'll be seeing a lot more studies that would support these initial results.
All right. Once again, thank you very much, Dr. Moran. I'm afraid that's all the time we have for questions for now. So we'll move on to the next portion of our program. The Philippine Genome Center prepared a brief video titled Bayanihan Hashtag Para sa Bayan, Isang Pasasalamat, to extend our gratitude towards all the individuals and groups who have assisted PGC through their generous donations. This also includes our thanks to the whole PGC community and staff for all of their hard work. So let's watch the video. Thank you once again to the whole PGC community, as well as our very generous donors. For our next talk, let's welcome the laboratory manager of the Clinical Genomics Laboratory, as well as the Clinical Health Officer of the Philippine Genome Center, Dr. Mark Edsel CAI. His presentation is titled Managing a Pandemic, Insights and Learnings from the PGC Clinical Genomics Laboratory Experience.
All right, good morning. So thanks for coming. So I'm Dr. Ayes. I am the laboratory manager and clinical health officer of the Clinical Genomics Laboratory of the Philippine Genome Center. And I'll be discussing my presentation entitled Managing a Pandemic, Insights and Learnings from the PGC Clinical Genomics Laboratory Experience. So the Clinical Genomics Laboratory was actually envisioned early on in 2018 and eventually into 2019 to be sort of the hub that integrates the scientific side, which is the services and the expertise is developed by the DSCF, the DNA Sequencing Core Facility, and the Bioinformatics Core Facility, into the clinical sphere. So tapping into our expertise in the Philippine Genome Center in a way that can benefit patients and clinicians in terms of world-class molecular diagnostic service. Now, fortunately or unfortunately, sorry, before that, and this is actually the broad range of services that were initially planned, starting with cancer, which is one of the forefront and needs, especially in the clinical sphere in terms of molecular diagnostics, and where a lot of studies are coming up and showing clinical benefit for patient populations, but as well as emerging fields in molecular medicine, specifically in cardiology, diabetes and metabolism, pharmacogenomics, and most importantly, also mental health. But again, as I was saying a while ago, fortunately or unfortunately, there was a pandemic that occurred in 2020. So the coronavirus 2019 virus had brought to our attention the need to reorient our services, especially since we were in the planning stages of launching it this year. But because of a specific call to action in order to serve the needs of the nation, or in this case, RT-PCR molecular diagnostics for diagnosing the disease on a very wide net of operations, we had to respond by shifting our services to do RT-PCR testing for coronavirus. And in effect, and this is why I said it was fortunate despite the unfortunate events of the coronavirus, it helped jumpstart the clinical operations of the laboratory and at least give us some sort of name. Because now, because of our operations, many people are now familiar that the Philippine Genome Center exists and that we provide clinical services based on the expertise that has been developed in this research institute. So just a brief story or history of the path that it took to come here. We actually began our services quite early on in the pandemic. Pandemic was first announced in March, and in April, we already were moving into getting our LTO as a special clinical laboratory. But with the mandate that we would start COVID-19 testing, we also did alongside that our accreditation for COVID-19 testing. So as early as the very start of May, we already started receiving clinical samples. And this was a gradual build-up because our laboratory did not have clinical staff. It was first run by scientists, especially from our DNA core sequencing facility, which I'm very grateful for their time and patience in training some of our clinical staff that we eventually hired over a transition period in June. Until July, we were finally able to work independently as a laboratory within the PGC framework. So to give you a context of how we operated in the Philippines, in April, when just a month after the pandemic occurred, as you may know, there were first 4 subnational laboratories that were recognized at the very start of the pandemic. But as soon as 1 month into the pandemic, there were already 24 licensed laboratories. And you can see here that they were averaging around 3,000 to 5,000 tests a day only. And the average turnaround time could be as long as 2 weeks. Near the peak of the pandemic in around July, so this is around 3 to 4 months after the start of the pandemic, we see an increase in about 254%. So now there were 85 laboratories. They were testing 4 times as more as they were at the start of the pandemic. And the average turnaround time was now around 34 times faster. So they could turn around 2 to 3 days. It was around this period that we were also ramping up our operations. And now, as of November 2020, there are now 186 licensed laboratories. So the DOH has been licensing around 23 laboratories a month, and there are still many laboratories pending accreditation. And on average, the Philippines is now testing 40,000 people a day, with a stable turnaround time across the different institutions of around 48 to 72 hours. For us, we started in April, of course. As I mentioned before, we only had 8 molecular biologist staff, assisted by members of the NIMBB of 12. There were 2 doctors, me included, and our molecular pathologist, Dr. Hui. We had 2 PCR machines, only 1 RNA extraction room. We were averaging around 134 at the time, and our average positivity was quite high, actually at 15.49. And we were serving 26 hospitals. At the peak, we were able to receive some support from the DOH, and we were able to fund for the hiring of 20 medtech staff thanks to the DOH PGH. We were also able to get data encoders, additional PCR machines. We set up a second RNA extraction room to handle the additional load. And because of that, we were averaging around 316 samples a day. Our average turnaround time also was able to meet the national standard of around 48 to 72 hours. And we were serving around 40 hospitals and healthcare units. As of today, we currently employ 31 medtech staffs. We have additional SRAs, scientific research assistants, which is an additional endowment from the DOH as a part of the support for our biosurveillance efforts, which we have been duly deputized by the department. And on top of that, lab aides and technicians, administrative officers, and additional doctor staff to help with the laboratory. So as you can see, there's been a lot of support in the COVID-19 testing facility of the Clinical Genomics Laboratory throughout the pandemic. So at present, we are doing 24-hour operations because of the number of staff that we have, with an average turnaround time of 24 to 48 hours. We conduct COVID-19 research and biosurveillance thanks to the addition of the SRA staff. We also have elective swabbing services, so we're already able to provide swabbing services to the community who would need it for purposes of clearance, for example, or travel. And we are already embarking in collaboration with the UP Diliman and the UP Health Services mass community screening effort. So this is our current running average. We process 258 samples a day on average, with a theoretical capacity of up to 800 specimens we can test in a day. So this is a schematic of our elective swabbing. We are actually granted through, thanks to the UP Task Force on COVID-19, Diliman. We were allowed to use the parking lot of the Institute of Mathematics, which is very close to the CP Garcia exit, corner Katipunan Avenue. And it's been in operation since October 2020. Our prices, you can see it here, which is actually aligned with the recent JAO regarding the price ceiling for testing, which is 3,800 pesos. We can do up to 100 tests a day, but we average around 20 to 30 based on the inquiries that we receive for testing. The UP Diliman is also embarking on mass screening, which is in an effort to secure both the safety of the community and as well provide information that will help in biosurveillance and also planning and policy development, especially now that we are reaching a point in the pandemic where we are rounding the curve, so to speak. Now more than ever, we are encouraged to do screening programs so that we can prevent any possible outbreaks, or at the very least mitigate the effects of a potential outbreak, which we should anticipate can happen over the course based on our historical experience with other pandemics such as the 1918 Spanish flu. So our target right now is mostly the key employees, UP employees, and also contractual workers and other essential workers that enter the campus, with the goal of identifying any hotspots or predicting the movement of certain strains of viruses through combined RT-PCR diagnosis and next generation sequencing. So it'll help give us an idea of what is the current circulating strand and any understanding also of the transmission patterns and also ways of us for which we can anticipate possible outbreaks based on the currently circulating strand. So we're planning to target around 3,000 to 6,000 individuals, hopefully by the end of this year. Now, and that brings us to actually how we're doing in terms of all of the samples we've processed. So this chart here just shows you the number of samples that we have processed over a period of 7 months. And you can see the general increasing trend. In July, we peaked because that was when we were given most of the additional staff to increase the capacity. And it was around also this time that you can notice the highest spike of the positivity. So the red line overlapping the bar graph represents the average positivity. And we actually peaked at around 38% highest around August, which coincides with the national peak or spike during the pandemic. In September, we experienced a drop because of some issues in the laboratory due to and requiring decontamination. But by October, you can see a general declining trend also with the positivity rate, which is aligned with the national recorded average that's occurring. Right now, our average monthly positivity rate is around 6%, which shows that we are seeing a phase in the pandemic where we are slowly seeing less and less cases. Now, in terms of testing, we partner with 47 different healthcare units, either government hospitals, private hospitals, city health offices, rural healthcare units, and others. And in this case, the others represents our own testing facilities and other elective screening subgroups or units where the patient population are asymptomatic people, but we engage in doing asymptomatic testing for screening purposes. So you can see a bulk of our testing is for the symptomatic patient population, and we have done up to 39,858 tests with a total unique positive case count of 7,304 individuals, majority of which coming from the city health offices, primarily because one of our major partners during the peak of the pandemic was the Quezon City Health Department, which is we are a constituent of the Quezon City. But when combined with the hospitals, they roughly make up half and half of most of our operations. And you'll see also in the distribution of cases, the positivity rate across the symptomatic spectrum is relatively the same, with no specific unit contributing more to positive cases than the others. Yung other section here, the fifth bar here, shows the asymptomatic population, which has a positivity of 5.2, which is actually uh, concordant with the national positivity rate as shown in the latest update by the World Health Organization, where they say the positivity rate is around 9%. Now, of interest here, at least, is the rural healthcare unit, actually, based on our information over the course of the weeks, shows the greatest relative increase in positivity despite the decreasing trend in positivity across the country. So, in terms of the data that we have, which may or may not be representative of the national picture, the rural healthcare units outside of the major metropolitan centers might be sectors where we want to pay particular attention to possible outbreaks that may occur, just based on the slight increase in positivity over time relative to the other cases. In terms of gender, we are similar to the World Health Organization skew. You'll notice that majority of cases are in the working age population, which is around 20 to 39 years of age. In particular, our statistics show a greater number of people from the 30 to 39, while the World Health Organization shows a larger proportion, the 20 to 29 population. This may be reflective of the general demographics that we see, specifically because they are hospital-based patients. But we can expect in a mass in a wider population network, especially as documented by the World Health Organization, that you would expect to see more of the non-working population, or sorry, the younger demographic population to be represented. Either way, in terms of male to female, there is no specific predilection towards either male or female, with a more or less even spread. Males are about 55% of our population, showing a general equal distribution between male and female in terms of positivity. Of note, we have the youngest, of course, in the range, of course, of testing is with the youngest of around 1 day, so prenatal, and the oldest is 119 years old. So there's a very wide spread of ages in terms of what they're covered. But in general, this trend is consistent with what we see in the national statistics. So, of course, aside from the testing, the clinical genomics laboratory is also engaged in research. Since we have been biobanking samples since the start of the pandemic, and there have been some calls for research utilizing some of these samples that we have banked in our biobank. Of two of important note, which we are currently working on, are viral genome sequencing of repeat and persistent positive cases. In literature, there have been questions about the persistence of positivity in certain patients, and a recent Nature correspondence where there was a immunocompromised patient with documented repeat infections, and they had a very nice phylogenetic analysis of the mutational changes of the virus throughout the course of the patient. So this would be an interesting thing to look into if we can actually sequence the viral sequences of these repeat positive cases that we have documented in our biobank. And secondly, we are like I mentioned earlier, with the mass screening of the UP Diliman, we will also be doing next generation sequencing as part of our continued biosurveillance effort. And the future direction for the Clinical Genomics Laboratory, of course, we are reorienting again for 2021 towards the core functions of the laboratory, which is to provide clinical diagnostic and molecular diagnostic services. Particularly, our first target is in the field of molecular oncology, where the demand is notably is there. So, along with this, it will be the development of our cancer genomics program, where we will begin biorepository of yung mga clinical genomic samples and sequences of patient cancer lines, and also biobanking for future cancer line development. And along with this, with some of the medical technology staff that we will be retaining, there will be a increased focus on staff training and capacity building to align themselves with these next generation sequencing modalities. So that's it for my talk. But to end, I'd just like to say special thanks, of course, to the Philippine Genome Center family, particularly our executive director, Dr. Saloma, the head of the DNA sequencing core facility, Dr. Maralit, and the core facility for bioinformatics, Dr. Ayap, as well as our other administrative staff in the Genome Center. Of course, UP Diliman, with their immense support for the programs and the development of the COVID lab, especially our Chancellor, Dr. Fidel Menzo. Department of Health, of course, Dr. Duque, Dr. Vergere, ASEC Santiago, and also ASEC Mercado Grande. Um, my staff at the Clinical Genomics Laboratory, there are a lot of you. So there they are, they're in that picture there. The UP Foundation, the NIMBB of UP Diliman, and also special thanks to the support of my clinical mentors and also mentors in the CGL, Dr. Eva Cochonco de la Paz and Dr. Raul Testora. And with that, thank you very much for attending this talk, and hope you have a great day.
Thank you very much, Dr. Ayes, for providing all of those invaluable insights. And now, once again, we'd like to solicit questions from the audience for Dr. Ayes. I believe we have some here. One was actually submitted for Dr. Meredith, but I believe Dr. Ayes, you are well suited to answer this question. Could you please shed light on the different available tests and what is the best test to determine if one is positive or negative?
Okay. Hi. So, good morning. First, apologies. I'm wearing a mask because I'm inside the laboratory, so our biosafety requires us to wear a mask. But thank you for the question. That's a very important question because, as many people know, there are a lot of in terms of the approved tests for diagnosing in general, you have your RT-PCR based technology. So they can either be in the form of what we do, the traditional RT-PCR, or what you might have heard as the rapid or gene express. These are both RT-PCR based and they are considered gold standards. The only difference between the rapid RT-PCR gene expert is that you can get the result usually within 24 hours. So they're ideal for stat testing. So for example, we get a lot of cases where, some people send us samples. We don't do the rapid RT-PCR, we do the standard, but then they ask us, kaya bang istat? Sometimes we have to make concessions, but ideally it would be best if stat procedures would be submitted to laboratories that have the gene expert module. So when in doubt, there is a DOH mandate that certain laboratories that have gene expert machines, specifically the TB dot centers, because they have the gene expert module kasi is also used for diagnosing TB. So it's just a matter of using the cartridge for COVID-19. Those are the ideal laboratories to send stat testing. Now, I'm also sure that you've heard of the other rapid tests. So there's the rapid antibody and the rapid antigen. So rapid antibody is intended to detect previous infection. So we use it in the clinics to determine whether or not a previous bout may or may not have been um, COVID-19. So it's only useful to detect possible previous infection. In cases when I'm treating a patient, for example, it can be used as a surrogate to determine the course of the patient. Like, for example, if they're hospitalized and they're still symptomatic despite being hospitalized for several weeks, you can actually utilize the antibody test to determine whether or not they have seroconverted and have circulating antibody. So that's one of the uses. But as a screening tool, the rapid antibody test has already been superseded by the antigen test. And the rapid antigen test is intended to detect circulating virus early in the case. Because what you're detecting here is the actual antigen or yung parts ng virus. So you see this much earlier in the infection. The only caveat here, and this is kind of hard to explain to many people, is that it is reliable in the context of what we call a high pre-test probability. What that means is if the context of when you're using the test is number one, there is a high positivity in the population of which you're testing, meaning the context, like for example, an outbreak. And number two, the patient has symptoms to which an exposure to which the clinician is considering highly the possibility of COVID-19, the results of an antigen test may be considered. So, for example, if I test someone na halimbawa height of the pandemic, na may symptoms siya and may possible exposure, I can do an antigen test. If it turns out positive, treat as if positive. If negative, I do a confirmatory. But if those conditions for high pretest probability are not present, the antigen test will require, whether or not it's positive or negative, will require a confirmatory RT-PCR test. So the bottom line is, RT-PCR test is still the gold standard. The antigen test may be used in the context of screening, but we recommend it be used by specialists who are familiar with when to use confirmatory tests to confirm the diagnosis. And lastly, the antibody test be utilized only during the course of the infection to determine historically the presence of an infection.
Thank you very much, Dr. Ayes, for that very comprehensive answer. I think we have another question. So, based on the information the laboratory is currently obtaining, in the recent report of a new record number of deaths in the country, do you anticipate an increase or second wave moving forward? I think this is a lot of people's minds, so we'd appreciate your insight on this.
Okay, so of course, no. As much as the problem when we make predictions is sometimes they're wrong. No, just like wrong. Work is of course in the 1918 pandemic, for example, there were actually two waves. There were three waves. Two of them were resurgences after the first wave. They all occurred over the span of around 1 to 2 years after the pandemic. Now, the reason for those is, of course, a combination of factors. But one belief, of course, is that there is a mutation that occurs at some point which increases the infectivity of the virus. This, of course, has not been documented. It's theoretical. And that's also one of the reasons why, as Dr. Maralit has mentioned, it's important for us to do sequencing during these times of the pandemic so we get an idea of what the circulating strain is and whether or not this confers increase in infectivity. The other factor is also the human factor, no? As much as, and the human factor has a lot of other factors, aside from the fact na syempre, alam ko lahat tayo pagod na, no? It's very tempting and we see it around now. A lot of people are starting to leave their homes. They're getting comfortable with the idea of wearing a mask and face shield. But again, with more human interactions outside, the risk for transmitting the virus becomes ever so much higher. And especially since we are reaching the holidays, I'm pretty sure lahat tayo gusto magsalo-salo sa Buena Noche and all that. But again, it goes without saying that we cannot predict when a second outbreak may occur. But just by looking historically as what's happened in the past and even in our neighboring countries, we should always be prepared for the possibility of a second wave from occurring, which is why the efforts of the Philippine Genome Center has been to promote active screening of asymptomatic populations and biosurveillance through next generation sequencing in order to mitigate or lower the effects of a potential second wave from harming economically, socially, any the country in the future.
Thank you very much, Dr. Ayes. I'm afraid that's all the time we have for questions for now. But I want to assure all of our attendees that our panelists will do their best to answer the questions on the Zoom chat. So now, before we proceed with our last main talk for the day, we have one more message from a distinguished guest. It's my honor to introduce the Secretary of the Department of Health, Dr. Francisco T. Duque.
To Secretary Fortunato T. de la Peña of the Department of Science and Technology, Professor Prospero P. de Vera III, Chairperson of the Commission on Higher Education, UP System President Danilo L. Concepcion, Dr. Cynthia P. Saloma, Executive Director of the Philippine Genome Center, Dr. Raul V. Dastura, Deputy Executive Director of the Philippine Genome Center, Dr. Benedict A. Maralit and Dr. John Michael C. Yap, Program Directors of the PGC. Dr. Mark Edsel C. Ayes, Laboratory Head of the PGC's Clinical Genomics Laboratory. At sa lahat ng atin mga kasama ngayon, isang magandang araw sa inyong lahat. Noong simula ng pandemya, tayo ay nangapa sa dilim. Subalit dahil sa walang humpay na pagsasaliksik, kasipagan at husay ng ating komunidad ng mga siyentista, unti-unti natin naunawaan ang COVID-19. Ang mga pananaliksik at pagsusuri ng ating mga eksperto ay patuloy na nakakapag-ambag sa yaman ng kaalaman tungkol sa pandemyang ating hinaharap. Maliban sa pananaliksik, ang Philippine Genome Center ay naging katuwang natin sa pagpapataas ng ating testing capacity sa buong kapuluan. Higit pa rito, ang pagbibigay ng PGC ng patnubay sa iba pang mga laboratorio sa pamamagitan ng best practices at process refinements ay lubos na nakatulong sa ating pagtugon sa COVID-19. Ang mga aral na dala ng COVID-19 ang siyang gagabay sa atin upang mapaunlad ang ating public health system at upang mabisang matugunan ang mga panganib na dala ng emerging at re-emerging diseases. Kabilang dito ang higit na pangangalaga at pagpapayaman sa larangan ng agham. Lubos ang aming pasasalamat sa PGC at sa komunidad ng ating mga siyentipiko at eksperto sa inyong pakikipagbayanihan para masupil ang COVID-19. Sa patuloy na paggabay ng scientific community, tanglaw ng kaalaman, ako ay panatag na tayo ay mananaig sa COVID-19.
Thank you very much, Secretary Duque. And next, originally we had scheduled a message from the chairperson of the Commission on Higher Education, Professor Prospero P. De Vera III, but unfortunately, due to other commitments, they're not able to participate this morning, although they do send their apologies and regrets. So instead, we'll proceed with the third main talk for this morning's event by the Program Director of PGC's Core Facility for Bioinformatics, Dr. John Michael C. Yap. We will be speaking on computational considerations for biosurveillance and genomic epidemiology. Dr. Yap, you have the floor.
So, good morning, everyone. So glad of you to join us today. I'm here to talk about computational considerations for biosurveillance and genomic epidemiology. So here's the outline of my talk. And let's first start with the what's and whys. So what is biosurveillance? Is it biologists playing Big Brother on all of us? Not so. It's a field, or it's also a process that detects and characterizes outbreaks of diseases in living beings. More so, it monitors the environment of causative biological agents that cause the disease. As a process, it involves the systematic collection and analysis of data for detection of cases, of outbreaks, and of environmental factors affecting the disease. Okay. How about genomic epidemiology? Is it Philippine Genome Center playing, Plague Incorporated on the Filipino populace? Again, not so. Okay, so it's basically an integration of epidemiologic methods with principles from genetics and genomics analysis, right? While biosurveillance focuses on any living beings, the focus of genomic epidemiology analysis is usually human disease and the role genes play in susceptibility or immunity thereof. Okay. That said, genetic and genomic analysis can focus on either the host or the pathogen, or possibly both. Now, why do we have to put some time and effort into this field or these fields, right? So, as we've seen in the ongoing pandemic, disease outbreaks are silent, and more often than not, they deal damage before they're even detected. And when they inflict this damage, they do so quickly, and as such, the spread is also as fast. Furthermore, to be able to counteract the effect, the window of opportunity to limit or mitigate its effects can only be as brief as a few days. Now, why do we integrate genetic and genomic analysis in this case? Because we want to underpin the genetic mechanisms to properly design diagnostic and therapeutic solutions. So with the basics out of the way, let's now move to a computational framework for biosurveillance and genomic epidemiology. Let's first start with biosurveillance. Here in the schematics, you can see that in the process of doing biosurveillance, you have several data sources from clinical and environmental monitors that are collated in a database, of which the data of which are then subjected to analysis to be able to detect outbreaks and even small cases, and then subjected further to other analysis to identify other causative means or causes, as well as other patterns that may emerge, which are then seen by clinicians or decision makers so that they can make proper or design proper response systems to mitigate the outbreak or disease. Now, in terms of computation, the focus of that would be these two middle parts, particularly in data collection and in the data analysis part. Another view or another framework which we could check here is in genomic epidemiology. Here, sequences sourced from infected patients are subjected to bioinformatics analysis and integrated with other known informations regarding the pathogen, okay, so that patterns, information, and even solutions can be derived or inferred. The results of the analysis are also made available to different levels of consumption, from the very technical to the very practical. So now, given these two frameworks, we could actually take out some of the similar features of the frameworks to come up with so-called unified computational framework. So here we have a database or data store which stores the raw sequences, raw sequence data, as well as other clinical and metadata regarding sequences, which can be submitted to an analysis pipeline, okay, the information of which can actually be stored back in the database so that it can be used for further analysis. Now, that's on the back end of the computational system that we're trying to come up with. For the front end, particularly for the client side, right, you have here an interface which can directly interact with the database for certain data transactions, or can interact with the analysis pipeline for the user to be able to conduct their own analysis. So based on this computational model, we will now identify certain requirements and tasks for biosurveillance and genomic epidemiology. Now, for the database, we first consider hardware, okay? So of course, the most fundamental would be storage capacity, okay? Now imagine a specimen, okay, when sequenced can churn out up to 2 gigabytes of data. And of course, you need to come up with probably hundreds and even thousands of specimens. So you have to consider storage capacity that can handle terabytes and even possibly petabytes of data when you've already collected a lot more samples, okay? And this is not even counting the clinical and metadata that's available. Next is network capacity. Why? Because more often than not, transactions with the database, whether input or output, will be usually via network. So you'll have to consider bandwidth, so, and put sufficient bandwidth, okay, connected to the data store so that the transfer of data would be as seamless as possible. And then you also have to consider I/O latency, so input-output latency. Why? Because if you've seen a while ago in the framework, the interface can directly transact with the database or the data warehouse, okay? So which means that the data in the storage must be submitted to the memory, which can then be processed for return of query or for processing of modifications of the data. Hence, you need to consider internal input-output latency to make the transactions as fast as possible. Finally, you also have to consider archive and backup in case of disasters or calamities, or just archiving data which cannot be just disposed as easily. Now, for the software, okay, first also you have to consider the data model. What are the types of information that you will be needing for the analysis that you will be storing in your database? Next is optimized CRUD capability. CRUD stands for create, read, update, and delete, which are the 4 basic transactions that you could do with the database management system. So you have to make sure that the CRUD operations are as seamless or as fast as possible. And of course, you have to consider computer security, right? Particularly to make sure that there are no leaks or no compromises that can occur. So you have to consider encryption algorithms and even authorization mechanisms or access control and other security protocols that come. Now, for the analysis module or pipeline, for the hardware, of course, you have to consider core processors for the data processing mechanism to make it as fast as possible. The amount of memory, because any data processing that occurs, right, has to be loaded into memory. So you have to make it as big as possible, especially when you have to do very intensive data processing. And as with the data store or database, you have to consider I/O latency, particularly when transferring data from your storage to the memory. Now, for the software, there are 2 things that you have to consider in terms of the analysis that you have to do. So first is the basic analysis, okay, which involves quality control of the data to remove errors, biases, or to come up with a minimum expected or sufficient data set for the analysis. Okay, you might want to identify which known pathogen sequences are most similar to the data or the sequences at hand, or to try to come up to measure the similarity or differences between the sequence data that you have. Sometimes you might want to see the entire picture, what the pathogen sequence, the entirety of it looks like, genome in particular, right? So you might have to do some assembly, and then you might want to identify mutation hotspots or variation hotspots at certain positions of the genome or the supersequence. And then finally, you might want to check the relatedness of your sequence vis-a-vis with the so-called family tree of the entire documented pathogen sequences available. So you have to take note that all of these actually are usually compute and memory intensive. So that's why you have to consider the hardware. Now, out of the basic analysis, you could do some advanced analysis. For example, you could integrate the results of your basic analysis with clinical data or metadata to infer hidden information or patterns that may emerge, and thus tying it with the disease outbreaks, for example. Okay, you could do spatial temporal analysis to be able to see information that may be tied to space or time consideration, or try to see how the pathogen or the disease outbreak spreads or evolves over time and space. And then, of course, one particular thing that a lot of clinicians or decision makers would want would be forecasting capabilities. So here you could add statistical or predictive models to be able to project or to somehow predict the spread of the disease or possibly the evolution of the sequence of the pathogen. Okay. Now for the interface, which now lies at the client side, so normally don't have control over the hardware specification. So we just have to consider some technical aspects. For example, you would want to have it as to consider or to be able to run in as many an operating system or even platform as possible, right? hence why a lot of interfaces are browser-based. As much as possible, you also have to make sure that it's not hardware intensive. Okay, and of course, you also have to consider the background of the users that will eventually tap into your system. So make sure that the user experience is also well considered. So for the interfaces that you need to develop, okay, of course, data management system to interact with the database, platform for data analysis from the very simple up to the very complex, possibly, and of course, a way to present your data in terms of non-textual, possibly information that could be easily digestible or where patterns can easily be inferable by way of data information visualization. So those are some of the considerations, basic considerations for the system that we need to design. Now, there are other considerations that I want to add in the design of the system. Okay, for example, availability of data. This refers to data sharing and openness and willingness of institutions to share the data that they have. Okay, as well as capacity of, for example, health institutions to actually generate the data and to store the data for later processing. Of course, since we're most likely dealing with humans most of the time, okay, ethical considerations in the way data is being extracted and handled should be considered, especially since the rights to privacy and information come into play. Now, of course, capability on the human resource end, okay, is also you also need to it also needs warrants look, particularly because you need people who are at least competent enough to be able to handle the analysis and to process the data and the output of your system. And then finally, digital inclusivity. You have to make sure that as much as possible, the design or your system isn't a barrier for, dare I say, less privileged researchers and technical people to be able to perform or to use your system. So those are the other considerations that come into play, and that ends my presentation. So thank you very much for listening. So I'll be taking questions and comments later.
Thank you very much, Dr. Yap. That was indeed a very informative and well thought out presentation. And once more, we'd like to invite our audience to please submit questions for Dr. Yap or our other panelists. And I believe we have one here.
Right. So, okay. Okay, is PGC currently doing biosurveillance and genomic epidemiology work, possibly for COVID-19? Well, as hinted a while ago by several speakers, of course, particularly Dr. Maralit and Dr. Ayas. Yes, we're doing, or at least Dr. Maralit is spearheading a biosurveillance effort for COVID-19. The bioinformatics team is still involved there to do the computational analysis for sequence similarity and phylogenetic analysis of SARS-CoV-2 sequences that are extracted and sourced here. In terms of genomic epidemiology, we're trying to lay down the groundwork to be able to do that, but we're also looking for additional funding. We're packaging it as a research for a proof of concept system and process to be able to conduct genomic epidemiology as applied for COVID-19. So there you go.
Thank you very much for that insightful response, Dr. Yap. Now we have another question that was submitted. With regards to the considerations on human capacity and digital inclusivity, how do you think this can be addressed in the country?
Okay, so first off, okay, I'm of the belief that biosurveillance and genomic epidemiology in particular are for the good of the country and the public. So that means the government should spearhead efforts to capacitate such efforts to be able to conduct endeavors in the fields. Okay, so now specifically for human capacity, okay, two things come to mind. First is to entice students and the youth, to at least a portion of them, to enter the field, to study the field, so that we could have a stream of experts that can actually perform the computational analysis needed for biosurveillance, or even the wet lab, the genomics or the biological aspects of it, right? But at the same time, there should be an environment that should foster or should be able to um, handle or welcome those stream of experts that will come into play. We cannot just have students who enter, for example, bioinformatics or genomic epidemiology or biosurveillance, graduating, then they don't have any careers or any viable work right here in the country, especially now that we've seen the need for it. So there must be a way to foster industry or careers in that kind of field. Okay. Now, for digital inclusivity, again, as I mentioned, government should be spearheading this. So, in effect, they should have support or some sort of plan to be able to enable as wide as possible, ideally all of the all Filipinos to be in a sense digitally, for lack of better term, savvy, or as part of to become digital natives in effect. So that means providing public infrastructure and even capacity building to be able to engage in ICT related services, or sorry, ICT related work or studies later on. So that is by far what I think would be the best way to foster digital inclusivity in this case.
Thank you once again, Dr. Yang. And I think there are several questions submitted. I think we have time for one more question. Somebody is asking, is the PGC on the lookout for looming spillover events in the future? Is this part of PGC's biosurveillance efforts? I'm not personally clear on what spillover means. They might be referred to super spreader events. In any case, we'd love to hear your insight, Dr. Yan.
Yeah. Well, that's why we started this. And in fact, as Dr. Morales has mentioned during his presentation, health-related emergencies and genomic surveillance of such is PGC's primary raison d'etre. So that means, we, as much as possible, we want to be able to conduct biosurveillance activities, not only for COVID-19, but for other future epidemics or outbreaks that may happen. Of course, that also boils down to the funding that we could receive, or the experts that or the manpower that we could have towards the future. So there.
Yes, I believe Dr. Salama wishes to answer one of the questions we received on Zoom.
So I just would like to,
Is-
yeah.
Yes,
So
please go
Dr.
ahead.
Carlo, thank you very much. I just would like to tell the audience that all the data that the whole genome sequence data at least are all shared through the GSAID website. So we have uploaded all those information. Regarding our analysis going forward, the project that has been funded by the PCHRD are all retrospective, at least for Dr. Maralit. So these are all biobank samples from the different SNLs. So going forward, I believe that the Department of the RITM will be also doing sequencing of prospective samples. So that is an information we would like to share with the audience. And also from us, from the PGC, from time to time, we will also do prospective cases as part of our duty, being deputized by the DOH for biosurveillance purposes. So that also, of course, will depend on whether we still have enough funds to do the whole genome sequencing going forward. Thank you.
Now we will be sharing a recorded message by Frances Fuentes Acosta on behalf of the Molecular Biology and Biotechnology Diliman Foundation Incorporated, or MBBDFI. This is a non-profit institution which has been instrumental to PGC's efforts during this pandemic. So let's all watch.
Hello PGC, and thank you for inviting me to give a look back on behalf of the MBB Diliman Foundation. So it was still the early days of the pandemic in the Philippines, 10 days after lockdown to be exact, when the foundation met online to discuss what we can do to support the Philippine Genome Center. So Dr. Saloma and her team had already identified the significant role that PGC can play in the testing efforts. However, budget and bureaucratic processes stood in the way. They still needed additional reagents, equipment, and accreditation to be able to get up and running as a fully functional testing lab. Now, we recognized the urgency, and the board quickly agreed to set up a donation drive to raise funds for COVID tests and related activities. Now, the ECQ restrictions, the inaccessibility of the Veterans Bank was just some of the many challenges that we faced. However, our members, our alumni, all threw in their support. And we all reached out to our networks to seek generous hearts through calls, text messages, social media. At the beginning, we were expecting mostly MBB alumni to donate. We anticipate maybe just a few 100,000, which would be equivalent to a handful of test kits or PPEs good for a month. But so what? That would still be something, right? And then yes, alumni did respond eagerly and enthusiastically. But not just them. We received donations from Filipinos here and abroad, private individuals, corporations, small businesses, even students. They donated money that they could easily have used for their personal needs during these uncertain times. Other groups had their own fundraising events like concerts and yoga classes, which reached even a larger network and contributed their proceeds to the foundation. So as word spread, other groups donated in kind, valuable PPEs, masks, and of course, the very much in-demand testing kits. So by the time we concluded, we ended up not with a few 100,000, but 3.6 million pesos. And from that amount, we were able to support UPNIH, UP Infirmary, the Lung Center, RITM, with majority going to PGC to provide them with the necessary lab renovations, to be a COVID testing lab, reagents, supplies, and testing kits. Now, I would like to take this opportunity to thank several people who made this happen. The foundation is not a very big group at all. We could not have done this without the board members and dedicated people who volunteered to help us. Thank you very much, Dr. Pia Bagamasbad and Dr. Neil Bascos, who selflessly devoted so much time, effort, and resources to make this initiative happen. Aside from our frequent operational discussions, they ensured that the donations were systematically recorded, accounted for, and disbursed. Dr. Ron D. and Dr. Charles Villamin helped me in assessing and approving the requests. Criselda Villegas set up the PayPal account and took care of transferring the funds to our bank account. Karen People and Jeliap set up and maintained the Facebook page, which became quite busy during the height of ECQ. We also had MBB volunteers, Wian Ibanez and Isabel Viola, who worked on the administrative but important tasks. Lastly, I would like to thank Dr. Saloma and the PGC for giving us the opportunity to collaborate and support you in the efforts to increase our country's COVID testing capacity. We are all aware of this valuable contribution and the service it has given the Filipino people. This pandemic has brought us so much tragedy, pain, and uncertainty. But through it all, we have come together to unite and show that despite seemingly insurmountable odds, we let science and determination triumph. I'm so proud to have been part of this effort. And despite everything that has happened, congratulations, PGC, and may we all have a Merry Christmas and a happy and a hopeful New Year. Bye.
And allow me to extend our thanks as well to Ms. Acosta and MBBDFI for that presentation, as well as for the incredible and inspiring work that your organization has done on PGC's behalf. And now, before we proceed with the final talk for this conference, we'd like to take a moment to show another brief video that highlights the theme of this conference. OnePGC, PGC in Luzon, Visayas, and Mindanao, hashtag para sa bayan. Aptly titled OnePGC in the Year of the Pandemic, this video features the Philippine Genome Center's efforts in addressing the national health emergency brought about by COVID-19, as well as activities and trainings conducted by PGC in Visayas and Mindanao. Let's all watch.
The Philippine Genome Center is a multidisciplinary unit in the University of the Philippines system, providing research, training, and sequencing and bioinformatics services. For over a decade, PGC has been leading the country's research and development drive in genomics and omics-driven technologies. Initially envisioned as a facility which will contribute to a timely and strategic response to threats of emerging infections, the unfolding events in 2020 prompted the Genome Center to utilize and optimize its resources to address the unprecedented challenges posed by the coronavirus pandemic. With its recently established clinical genomics laboratory, PGC equipped and reconfigured the lab to comply with the requirement of the Health Facilities and Services Regulatory Bureau of the Department of Health, and was certified to conduct testing for COVID-19 by real-time PCR. In coordination with the Department of Health, the COVID-19 testing lab started off as a non-hospital-based facility which accepted samples of priority cases for testing from hospitals, community swabbing centers, and other healthcare facilities in Manila and nearby cities and municipalities. To address the growing number of cases and contribute to targeted mass testing, PGC's Clinical Genomics Lab expanded its screening operations by opening a drive-through and on-foot swabbing area in the grounds of the Institute of Mathematics. In addition to its testing efforts, PGC fully utilized its DNA sequencing and bioinformatics core facilities in identifying nearly complete whole genome sequences of 6 SARS-CoV-2 from COVID-19 cases between March 22 to 28, 2020. It has since serviced the requests for next-generation sequencing of SARS-CoV-2 from other referral hospitals in the metro. To provide a broader view regarding the sources, spread, and evolution of COVID-19 locally, PGC is spearheading a national effort to do genomic biosurveillance of SARS-CoV-2 in the country, as the only local facility with a range of next-generation sequencers for a large-scale sequencing effort. Hand in hand with PGC Visayas and PGC Mindanao, NIH in UP Manila, and NIMBB in UP Diliman, the satellite facilities of PGC in Iloilo and Davao City have conducted trainings in molecular biology techniques, covering RNA extraction and RT-PCR for COVID-19 testing in the regions. These efforts are particularly important due to the constraints in inter-island travel at the height of the pandemic. The trainings have been a boon to local laboratories, centers, and hospitals as they prepare to be accredited as COVID-19 testing laboratories in their specific localities. These trainings have contributed immensely to expand the country's testing capacity at a critical juncture in our nation's fight against the COVID-19 pandemic. The Philippine Genome Center continues to advance its position as an institution on relevant multi-omics research and services that responds to the needs of the Filipino people, utilizing genomics for a better Philippines. Ang PGC para sa bayan.
Indeed, PGC para sa bayan. This video really highlights how so much has happened during this tumultuous year. Amidst all of these events, we hope to overcome the challenges before us and be able to continue to advance the vision of the Philippine Genome Center. And finally, to conclude this morning's event, we have a presentation on PGC's 6-year strategic plan, as well as some concluding remarks by our Deputy Executive Director, who spearheads the PGC's business development and resource generation. Let's all welcome Dr. Raul V. Destura.
On behalf of the Executive Committee of the Philippine Genome Center, allow me to share snippets of our game plan, which started way before the pandemic began. As a multidisciplinary research and service unit, we strongly support collaborative initiatives and multi-sectoral partnerships. As we reach out to researchers, students, colleagues, or different sectoral groups in Luzon, Visayas, and Mindanao, we are reminded of our mission to contribute to the macroeconomic growth of the country by continuously seeking new knowledge and innovations in the field of multi-omics sciences. At a glance, these are our goals. Position the Philippine Genome Center as an industry leader in big data analytics involving genomics research in the Philippines. Increase stakeholders' recognition and appreciation of genomics. Ensure operation efficiency as a research unit and service provider. Explore new business models and maximize existing ones for resource generation and program continuity. Establish new linkages and maintain collaborations with local and international institutions. Our situation may be a little bit complicated right now due to the COVID-19 pandemic. Almost all companies or institutions were virtually not spared from the after effects and ongoing effects of this pandemic. However, from the moment we as a team started in putting together this plan or blueprint or a whole gamut of a to-do list for the next 6 years, I know we can do it together. Our team, as reflected in this organizational chart, is a healthy mix of young and young at heart together that radiates vibrant energy and creativity, which makes this journey towards achieving our goals more exciting and fulfilling. And before we do the closing of this event, I would like to thank everyone who spent their precious time with us today, our key guests. Thank you for your messages and for supporting the Philippine Genome Center. Thank you, and as well as congratulations to our Executive Director, Dr. Cynthia Saloma, who was recently bestowed with the Giscoro L. O'Malley Award for Outstanding Science Administrator. And of course, allow me to applaud the entire Philippine Genome Center team for showing what unity and bayanihan truly means. Maraming salamat po.
Thank you so much, Dr. Destura. We are all looking forward to the Philippine Genome Center's future directions as OnePGC. To our audience, if you wish to view the full version of the PGC strategic plan, feel free to visit our website at www.pgc.up.edu.ph right after this conference. Furthermore, we encourage everyone attending to fill out the feedback form for this conference, which can be accessed at bit.ly/feedback_1PGC2020. The link is also shown on screen as well as in the Zoom chat. A certificate of attendance will be sent to you by email within the next few days once you've completed this form. Also, for other updates and announcements, please keep following PGC online at phgenome on Facebook and Twitter, and on our website at pgc.up.edu.ph. And with that, thank you everyone for joining the first PGC virtual conference, OnePGC, in Luzon, Visayas, and Mindanao. To officially end this program, please watch this short video on how to schedule COVID-19 testing at PGC's Clinical Genomics Laboratory. This conference stream will automatically sign off after this video. So once again, our thanks to all of you for attending, and we hope that you all have a good day.